Today we're going to look at the discovery of blood groups, specifically the ABO blood types. There were experiments for hundreds of years where we gave donor blood to recipients, but many of those patients died. It wasn't until we had more information, thanks to Carl Landsteiner's work, that it made it possible to determine blood groups and pave the way for blood transfusions to be carried out safely. For his discovery, he was awarded the Nobel Prize in Physiology or Medicine in 1930. He also helped Alexander Weiner discover the Rh blood type in 1937. And thankfully, we now knew about ABO blood groups. Again, that discovery by Carl Landsteiner was made in the early 1900s, and that made blood transfusions medically safe. But it wasn't until Charles Drew, an African American surgeon, developed the technologies to process and store blood that blood storage and blood transfusion technology really took off. Charles Drew was a pioneer in blood storage techniques, and he organized the first large-scale blood bank in the United States. But where does your blood type come from? Well, it comes from your mother and your father. You inherit one gene from each to determine your blood type. So your blood type comes from your genes. For blood type, we use the letter I to dictate between alleles. So you use a capital letter for the dominant trait and a lowercase I for the recessive trait. And these genes and how you inherit them in combination from your parents determine your blood type by causing proteins, surface proteins, called antigens, to exist in the cell membranes of your red blood cells. And so when we're talking about blood types of the ABO group, we're talking about blood types A, B, AB, and O. Those are the phenotypes, the physical characteristic, the blood type that you could have based on your genetics. So what is an antigen? Well, it's just a protein on the surface of a red blood cell membrane. An antigen can also be something foreign your body fights against. And because foreign blood can elicit a type of clumping or agglutination reaction, that's why these surface proteins are called antigens. They're also known as agglutinogens because they can initiate or stimulate that process of agglutination, the clumping of particles. The reason that the surface proteins on red blood cells can initiate an immune reaction that causes clumping is because our body comes with preformed antibodies, which are proteins made by lymphocytes in the plasma in response to the presence of antigens. So read that definition again. Proteins made by lymphocytes in plasma in response to the presence of antigens. So they're not normally preformed. They normally only form after coming into contact with something foreign that they don't recognize. But when it comes to ABO blood groups, we do have preformed antibodies against the blood type you don't have. And those antibodies bind to antigens on the erythrocyte surface and agglutinate them or clump them together. So let's review real quick. We have phenotypes and genotypes when it comes to ABO blood grouping. The phenotypes are O, A, B, and AB. And if you're type O or type AB, we know your genotype. It's only one option. If you're type O, you inherited 2 recessive alleles from your parents. And if you're type AB, one of your parents donated an A and one donated a B allele. But when it comes to type A and B blood, there's 2 options when it comes to the genetics that you inherited. You could be homozygous dominant, where you inherited 2 dominant traits of the same type, A or B, from your parents. Or you could be a heterozygote, where you only inherited 1 dominant trait and 1 recessive. And without molecular testing, we're not sure which one is which when you have type A or B blood. We can do test crosses or Punnett squares to kind of better predict the offspring from a parental cross. If you've never seen a Punnett square before, it is a box with 4 spaces, and you would put the alleles for one parent on the left and the alleles for the other parent at the top. And you bring everything on the left across and everything at the top down, and that helps you predict possible offspring from that parental cross. But what does this actually look like? What does it all look like when it gets down to the phenotypes? What is on the surface of the cell? Well, on the surface of red blood cells are antigens. So if you're type A, you have A antigens. Type B, you have B antigens. Type AB, you have both A and B antigens. And type O is kind of like type 0. You have no antigens. And then you would have antibodies preformed in your plasma against blood types you don't have. So if you're type A, you have B antibodies. Type B, you have A antibodies. If you're type AB, you have no antibodies because if you had A or B antibodies, they would attack your own cells. And if you're type O, you don't have A and you don't have B antigens, so you have both of those in your blood plasma to attack cells that do carry A or B antigens because you don't have those. So again, you have antibodies against the blood type you don't have. So with an A antigen on the surface, type A has B antibodies in the blood plasma because they should not recognize type B blood, but they have antibodies to attack it. Type B, you have B antigens, A antibodies. Type O, you have A and B antibodies, but no antigens when it comes to A and B on the surface of the red blood cell. Type O is also the universal donor, and as long as you donate packed red blood cells, this blood can go to anybody. And type AB has A and B antigens, and this blood type is the universal recipient because they have no antibodies because it would attack their own cells, but with no antibodies, they can receive any blood type, and we're not concerned with a transfusion reaction. All right, next we're going to look at how common your blood type is. If we count both positive and negative, type O is actually relatively common, but it is pretty rare to be O negative. It's actually more common to be positive than negative when it comes to blood type. And when we talk about positive blood, we're talking about blood that has an extra antigen that needs to be considered during a blood transfusion, which is a procedure in which blood is given to a patient through an IV line. And blood transfusions are done to replace blood loss during surgery or a serious injury, and it's done to make sure that a person's body can have enough blood, even if they can't make blood properly because of an illness. But we do want to match blood, both using the ABO blood groups and the RH blood groups, which dictate whether or not you're positive or negative. If you have the RH protein on your red blood cells, you're RH positive. If you're missing it, you're RH negative. And someone who is RH positive can receive positive or negative blood, but someone who is RH negative can only receive negative blood. And that's the clinical rule, because technically someone who is Rh negative can receive positive blood one time, but in a clinical setting, we don't know if there's been exposure to positive blood from an Rh negative individual. So clinically, we treat as if it's already occurred. So Rh negative can only receive negative, and Rh positive can receive positive or negative. What is Rh? Well, Rh stands for rhesus. It stands for Rh factor, or the D allele. It's an extra protein first discovered in rhesus monkeys, and now we know it's on human blood cells. And if your blood contains this extra protein on its red blood cells, you're said to be Rh positive. If your blood does not contain the protein, your blood is said to be Rh negative. So here are some examples of those blood types. If you're O negative, you don't have A or B antigens, and you don't have the Rh antigen. If you're O positive, you have the Rh antigen. If you're B positive, you have the B antigen and the Rh antigen. But if you're AB negative, you only have A and B antigens, no Rh. So when it comes to Rh factor or the rhesus factor, not only is it just a type of protein that's also or could be on the outside of your red blood cells, having it makes you positive, not having it makes you negative. So an Rh negative individual does not have a negative protein. The negative stands for the absence of the protein. Next, we're going to be looking at Rh factor in pregnancy and talking more about some interactions between Rh negative and positive blood.